So you guys, by now you should know what a bivariant is. We said it's a pair of homologous chromosomes. And like we said, homo means same or whole, same. So in this case, homologous chromosomes, they are chromosomes, two chromosomes formed from one chromosome by replicating. You need to know what homologous chromatids are. A moment. So let's proceed and see other important terms. Centromere, I've already talked about it, and I believe if you've been attentive enough, you know what a centromere is. But like we said, if I draw this as my chromosome, okay, you can guess where the centromere is. Try to guess. Where is the centromere? Try to guess. Yes, I hope you've guessed right. This point where the two chromatids join here, okay? It is the centromere, okay? And this one, we are going to use it also frequently. We shall use this centromere frequently for us to have that deep understanding of meiosis and mitosis. So if this is my chromatid and this is another chromatid, this point where the two chromatids attach, meet, join is known as a centromere. Okay? And still, if you want to separate these two chromatids, you still separate this centromere. And if you break the centromere, obviously, these two chromatids will detach from each other. So then another important term is the chiasmata. That's good. We shall also use that frequently. Now the chiasmata is, if I have, like I told you, if I have my two homologous chromosomes, that's chromosome A, okay? This is my chromosome A, and my another chromosome A, not so? My other chromosome, and it's another chromosome A. Now these two chromatids, these two chromosomes, they are the same. It's like this one has been replicated from this. They are the same. Okay? Yes. Now, in meiosis, we are going to see that these two chromosomes, they can pair up. They will occur in pair. Okay? They will attract each other, and then they end up coming close to each other, that their segments, these chromatids, they will end up coiling. Even here, this will end up also coiling with this, so that you form such a structure. You have this as your chromatid, and then this as our other chromatid of chromosome A of this chromosome, and then this one also will do the same. Okay, it will do the same. I'm trying to sketch. Even this one also will do the same. So my two chromosomes, these ones, they will come together and they end up exchanging these segments. Are you seeing? And they end up crossing. Now this point here, this point here, where these two chromatids, leave around this is the centromere. Switch, why do you want my screen control? So this is the centromere, okay? This is the centromere. And then this one is also another centromere. But this point here, okay, this one, where these two chromatids join, where the two chromatids join is known as the chiasmata. If it's one, it is chiasma. If they are many, they are known as chiasmata. Okay? Yes, this very word, this one. I hope it is really okay. I hope it is okay. In case it's not okay, you are free to ask. You are free to request, and I explain freshly. So you have your chromosome. This is chromosome A, and actually these two chromosomes are homologous chromosomes because they are formed from one chromosome. It's one chromosome that replicates, okay, to form another chromosome, and the two are the same. And after replicating, they will come close to each other and they end up crossing, okay? And the chromatids, they will end up crossing, okay? At the extreme ends, here and also here. Now this point where the two chromatids join each other, like how you can see, is known as the chiasmata, okay? In plural, you can call it chiasmata. If it's one, it is chiasma. Now, it doesn't stop at just crossing like this. No, it doesn't stop at just crossing like this. Somebody saying something. Does it also happen in mitosis? No, we don't have this crossing in mitosis. We are going to see. I like the insinuation. You are insinuated. Eh? You are curious, but have no worries. We shall see, okay? We shall see. But you keep the hype, okay? Yeah, keep the hype. So it doesn't stop at just crossing, crossing each other at this point, no. It goes ahead and the segments, these last segments of these chromosomes, this one, maybe let me sketch this with blue, okay? For easy understanding. So the whole of this chromosome, let me sketch it with blue. Okay? Let me sketch it with blue. A moment. Somebody is calling and I wasn't. Sorry for that. So I said they don't just stop at crossing, but they are going to exchange the segments. Okay? Where this one will break off and join this red chromosome, and this red will also break off and join this blue chromosome. And the same will happen. The blue one will break off from here and joins the red chromosome, and this red will also break off from here and joins the blue chromosome. So that you form your chromosomes as this. Are you seeing this one? Now, don't just look at this as simply segments. Don't just look at this as simply segments. No. You are exchanging DNA. You are exchanging genes. Okay? You are exchanging genes. Because these chromosomes, they have what we call the DNA. They are wrapped, okay, by the DNA. Are you seeing? Yes. So as a result, because you exchanged these two, we shall form different combinations. And these different combinations, they are the ones that will lead to formation of variations. You remember when we studied variations? You remember when we talked about the causes? You remember that we talked about one of them as crossing over? Now this is the process. This exchange of segments on homologous chromosomes, these ones, chromosomes the same, is what we refer to as crossing over. Okay? And it's really good. It is really good as far as variations are concerned. And it occurs in meiosis, and to be specific, in prophase 1. We are going to see all this. We are going to see all this. Also, guys, yes, anybody with a question, feel free to ask. Feel free to ask. Feel free to ask. Okay, guys? Yes. Somebody's saying, how can you define the segments? What do you mean by defining segments? How can you define the segments? What do you mean by defining segments? You can write on the screen somewhere here, or somewhere here, or you can still text. What do you mean by defining segments? Oh, the word segment has confused you. It's okay. The segment, I mean, if this is my chromosome, if I break it from here, this part, this upper part, it is a segment of this chromatid. That's what I was meaning. If I come and break it off from here, this part, this part, this one, it is a segment of this chromatid. So that's what I was meaning by segment. Yes, I hope all is good now. Okay, I hope all is good. Then another important term is haproid. Okay, I think I won't need this. Yes. So haproid means, let me explain it together with the diploid. I hope it's the next. Yes. Now, like I said, human cells are made up of how many chromosomes? They have 46. So a normal human cell has 46 chromosomes. This is the structure of a chromatid. Yes, yes. Simply, I didn't go deep, because if you go deep, you label loci and then alleles, but that is simple enough. Okay, for now. So now, a normal human cell has 46 chromosomes. Okay, 46 chromosomes. But these 46 chromosomes are arranged in pairs to form how many? 23 pairs. By the way, if you remember sex determination, we said that the 23rd pair is the X and Y chromosomes. They are the ones which determine sex of an organism. And somebody is asking, can they ask one to draw and label a chromatid? Yes, like I said, yes. However, on rare occasions, but anything is possible. Okay? Yes, they can, but it is simple. Just draw. Okay? Draw, but try to be neat. And then you label, this is a centromere, and then this is a chromatid. The whole of this is a chromosome. If you want, you can put the locus. Okay? And then in the locus, you put their alleles. And you say this is an allele. But it is simple, it's not hard. Okay? It is not hard. So now, we say that human cells have how many chromosomes? 46. That's brilliant. But the 46 chromosomes are arranged in how many? in pairs to form how many? 23 pairs. Pavin, why are you writing on the screen? And we said that the 23rd pair is the one which determines sex. This is actually the X, which is large, and Y chromosomes. Now, a cell with a complete set of chromosomes, all 46 chromosomes, is known as a diploid cell. But during fertilization, for instance, this cell with 46 chromosomes, it will divide by meiosis to form 4 daughter cells. 4 daughter cells. And each of the 4 daughter cells, somebody's saying, I was drawing something and show it to you. It's okay, you can draw it. You draw it here. You draw it here. I'll be able to see. So, like I said, if this cell, you have something to say? It's okay. So if this cell divides by meiosis, okay, it will produce 4 cells. We are going to see what meiosis is. What are you drawing? Let me watch. Yes, somebody is drawing a diagram which shows the stage of mitosis. Have no worries, okay? We are going to reach there and you will appreciate. But the key word is to first understand these key terms, okay? Yes. So have no worries, okay? Yes, all is going to be good. So just have a clear understanding of these terms, okay? Then we shall talk about meiosis, shall talk about mitosis and the stages. So, so diploid, if this cell divides by meiosis, okay, then it is going to produce 4 daughter cells, and each daughter cell, remember the chromosomes, the 46 are in pairs. So the chromosomes are going to be unpaired. And instead you have 23 chromosomes in each. Not pairs, but simply 23 chromosomes. Now this state where you have half the normal, half the normal, half the normal is known as haproid. I think ha means half. Okay, you get it from that. Then di means complete, I think in this case. So, but haproid means half. Okay? So the cell has half the number of chromosomes than a normal cell. Then we have replication. This is where a structure reproduces or produces an exact copy of itself. So basically, those are the terms that we are going to be using, that we are going to be singing as far as cell division is concerned. Now, cell division is divided into 2 types. Okay? We have mitosis and we have meiosis. Now, these 2 types, they are all processes by which a cell divides, okay, to produce other daughter cells. Okay? But they are all important in different aspects of life. Okay? Whereby in one, a cell divides to produce 2 daughter cells, and all the 2 daughter cells are exactly the same as the mother cell. But in another, you divide, or one cell divides to produce 4 daughter cells, and all the 4 daughter cells have half. Okay, this is half. If this is 1, if this is complete, if it's 1, these all the 4, they have half, 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 and half. Okay, the genetic material as compared to their mother cells. And all these processes, somebody is saying, why have you left replication? Okay, I've talked about it. I said process by which a cell divides to produce an exact copy of itself. I was illustrating it even during the entire explanation. So this half, 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 half. So all these 2 processes, they are equally important in aspects of life. So from what you have been discussed, right from genetics up to now, okay, which process is involved when a cell divides to produce 2 cells? Think about it. Think about which process, and then which process is involved when a cell divides to produce 4 haploid cells. Also think about it. Now, this one is mitosis, and in mitosis, 1 cell divides to produce 2 cells, and the 2 cells are exactly the same as the mother cell. You cannot separate, you cannot distinguish. You cannot, trust me, you cannot. The 2 cells are the same. Okay? They have same information, not just similar, but same information. And that's mitosis. But with meiosis, this one is found in reproductive cells. Okay? Reproductive cells. Such as production of sperms. I hope all of us here know what a sperm is, and we all know the structure and stuff like that. And then in production of ova. These are the female eggs. Also, meiosis is involved. Okay? Meiosis is involved. And you need to think about why. Before we even talk about, before we even describe how these 2 processes occur, you need to think about why. Should you produce a sperm cell by meiosis? Why should the sperm cell be half? And why should an ovum also be half? Whereas other body cells, for example, during growth, when you are growing, it is this process that is responsible. When you get an injury, when you get a cut, that after healing, that process of healing, you have to replace those dead cells. So think about why you use mitosis. So by the end of this chapter, you need to know why mitosis and why meiosis, and what mitosis is and what meiosis is. So now what is that animal called mitosis? Let's first talk about mitosis. What is that animal called mitosis? So before mitosis, there are phases, actually. There are phases. Now, all these stages, they are stages of cell division. Okay? But for a cell to divide and produce another cell, it passes through different phases, different stages, until when a cell successfully divides. Let me repeat that. If you have your cell, okay, for this cell to divide and produces 2 exact copies or 4 exact copies and stuff like that, it passes, this cell undergoes different phases from that, from that, from that, and finally you divide. The cell undergoes through different phases, okay? And in fact, in mitosis and meiosis, we are going to talk about these phases. And these phases are none other than, let's list them. The first phase, and this one is the same for both meiosis and mitosis. That's why it comes first before the other. The first phase is interphase. Actually, all of them have phase. So this is the very first phase of cell division. The very first phase of meiosis. The very first phase of mitosis. And it's the same for both meiosis and mitosis. Now from that, after interphase being the same for both, now the preceding phases, they'll be different for meiosis and for mitosis. Now the preceding phases, the second one is, can we guess? Yes, we have prophase. And then from prophase, we go to metaphase, and then from metaphase, we go to anaphase, okay? And then from anaphase, we finally end with telophase. Okay? So you see these stages, you need to understand what these phases are, the events that occur in each phase here, and the order, okay? And the order, okay? The order from bottom, top to bottom, middle, up. So the order. So we start with interphase, and then prophase, metaphase, anaphase, and telophase. If we start from down, it is telophase, anaphase, metaphase, prophase, and interphase. And interphase is the same for both mitosis and meiosis. We start from up again, interphase, prophase, metaphase, anaphase, and telophase. Start from the bottom, telophase, anaphase, metaphase, prophase, and interphase. Let's start from the second going downwards. Prophase, metaphase, anaphase, and telophase. And let's start from the second going upwards. Anaphase, metaphase, prophase, interphase. So you need to really know the order, okay? You need to know what these phases are, the events that take place in these phases, and the order of these phases. Somebody saying something? If you don't write them in the order, they become wrong. Exactly. This one cannot come before this. Okay? This one cannot be, no, that's madness. So you need to know them in their correct order. Let me try to rub and we do it together. Okay, starting from interface. We have interface. Prophase. Metaphase. Anaphase. And finally, telophase. I have some smart students here. Somebody has already developed an acronym. He's called, he's saying that P, MAT. This is being smart. Where this is prophase, and then metaphase, anaphase, and finally telophase. Yeah, that's good. That is good. Actually, that is one way of studying. Okay? If you can develop an acronym for something, it is okay. It is okay. Okay? That is good. Okay? Yeah. So if you can use his, it's okay. If you can develop yours, it is okay if you can. But the issue is the order. The order must be maintained. Somebody is saying Trump. Trump. So what is Trump? This is telophase. Anaphase. Metaphase. Prophase. And then interface. Okay, that is good. Trump from the back. It is okay. Okay. It is very fine as long as you develop it to help you memorize. Okay? The order, because the order is key. You lose out on the order, you lose marks. You won't know what happens under each phase and stuff like that. So I hope the order is okay. You can use PMAT according to him. It is okay. This is prophase, metaphase, anaphase, and telophase. Or you can use Trump, but Trump of A, not you. This is telophase, anaphase, metaphase, and prophase. And then finally, there is interface for all. Okay? That's brilliant.